Hi AP Bio students, so we're going to talk about mitosis and cell division. Excuse me. So we need cell division for three important reasons. The first one is growth. I've mentioned this in an earlier video, but a common misconception is that in order for us as humans to grow in size, our cells don't expand exponentially or just increasingly expand forever. Our cells just become more plentiful. Our cells divide, and that's what makes us grow. There's a bigger quantity of cells, not that the cells themselves expand and get larger, because that's actually not good for the cell to be super huge because of the surface area to volume ratio. The surface area to volume ratio basically says that it's more efficient for a cell to be smaller because that means you have more surface area and less volume, because when a cell gets bigger, the surface area and volume increase at different rates. So that's why you want to keep your cell size small, because then you have more surface area and less volume. And if you have more surface area, you have more area to exchange materials with the outside world, such as heat. You can exchange heat, food, waste, etc. So we need cell division to grow, and this occurs in multicellular organisms. We also need cell division to repair. Excuse me. Let's say you get a cut on your hand, like a paper cut. Obviously, it's not going to be there forever. The skin cells in your hand are going to receive a signal to divide and repair that paper cut. And in unicellular organisms and in multicellular organisms, we use cell division for reproduction. So some important vocab. This will probably come up on the AP Bio test, binary fission. I feel like that was on one of my multiple choice questions. So binary fission is bacteria cell division. Remember, bacteria are single cell prokaryotes. And prokaryotes, remember, they came before eukaryotes. They don't have a nucleus and they don't have other membrane-bound organelles. And so basically when bacteria divide, they're creating a whole new bacteria, since bacteria are only made up of one cell. Excuse me. And then we also have mitosis, which is the nucleus division in non-sex cells. So non-sex cells are also called somatic cells. These are basically just bodily cells that are not for human reproduction. And mitosis, a key thing to remember is it's just for the nucleus, it's not for the rest of the cell. And meiosis is basically the same thing as mitosis in terms of definition, except it's nucleus division in sex cells. So this is used for human reproduction. And we have cytokinesis, which is division of the cytoplasm. And the cytoplasm is basically the rest of the cell that's not the nucleus. So the cell cycle. The cell cycle tells a cell's life from formation to division. So we have two main parts in the cell cycle. We have M phase, this little part right here, which occurs at the end of the cell cycle. And then we also have interphase, which is the rest of this. And interphase is broken up into three main sections. The first one occurs right after a cell divides. This is called G1 phase. This is the primary growth phase of a cell so that a cell can grow to its regular size. And then we have S phase. S phase is very important to know for the AP Bio test. It came up in a lot of questions for me. And this is basically where DNA is replicated in your cell, in the nucleus. And then you have G2 phase after S phase. And this is where your cell gets even larger. It gets bigger so that it can prepare for cell division. And then we have cell division, which is M phase, which consists of mitosis or meiosis, depending on which cell you're using, and cytokinesis. So now I'm going to talk about mitosis. So first we have prophase. And in mitosis, there are 5 main phases: prophase, prometaphase, metaphase, anaphase, and telophase, or telophase, however you want to say it. So prophase is when the nuclear envelope breaks down. We have something called centrioles, which move to the poles of the cell, so the opposite ends of the cell. And centrioles are groups of star-shaped microtubule arrangements, and they're in something called a centrosome. Centrosome is a membrane-bound organelle that's home to these things called the centrioles. And centrioles make spindle fibers, and spindle fibers are basically these extensions coming out of the centrosomes. They kind of look like spider legs to me. And then after that, we have prometaphase. This is when the nuclear envelope finishes dissolving, chromosomes finish forming, centrioles reach the poles and form spindle fibers that attach to the centromere's kinetochore. So a centromere, okay, so basically this is a drawing of a typical chromosome after S phase because the DNA is replicated. And this whole thing is a chromosome. This center area right here is called the centromere. And in the centromere, there's a protein matrix called the kinetochore, which is where the spindle fibers attach to. And the spindle fibers are kind of these spider-shaped extensions right here. They kind of look like spider legs to me. And in a chromosome, here we can tell it's duplicated because we have not one sister chromatid, but two sister chromatids. And the sister chromatids are identical. So if you were to split a chromosome into two, you'll have two sister chromatids in this chromosome. And then after prometaphase, you have metaphase, which is where spindle fibers contract and pull chromosomes to the middle of the cell. And the middle area, so this, if I were to draw an invisible line in the middle of a cell, the middle area is called the equator or the metaphase plate. So now think M for middle. All the chromosomes are lined up in the middle of the cell. And this is probably the most easily seen phase under a a microscope, in my opinion. And then you have anaphase, which is when spindle fibers shorten, pulling sister chromatids to the poles. And now these sister chromatids, which are separated from one chromosome, are now considered separate chromosomes. The reason why when you separate the sister chromatids, now they're considered two different chromosomes. The reason that is because chromosomes are counted by their centromere. So this is one chromosome because you have one centromere in the center right here. And this is also considered one chromosome. Even though this looks like a sister chromatid, it's considered one chromosome because it has one centromere right here. So finally, you have telophase and cytokinesis. In telophase, the nuclear envelope reforms, spindle fibers break down, and chromosomes disappear. And in cytokinesis, that's when the cytoplasm divides. There's something called a cleavage furrow in animal cells that form to help pinch a cell into two, or something called a cell plate in a plant cell forms to help divide a plant cell into two.